My talk today, I gave a new title for my talk than the one that's on the program, and it's called The Infrastructures of Comparison, Collecting, Comparing, and Computing. So I know about your project about comparison, and I mean, 10 years ago, I wished so much there was such a project which I could have learned from, because as you've certainly realized, there was very little literature on this very important intellectual tradition of comparison. Which had been, I thought, understudied. And so it's wonderful that you are been working now for several years on this. I'd like today to tell you a little bit about my last book called Collecting Experiments, which came out already now in 2019 at the University of Chicago Press. And I will highlight what the main arguments are and the context for the book, especially for those of you who are not historians of science or even historians of biology, just that you can understand what are the main arguments here. If you're tweeting, you can always tweet using this handle. This is the handle of my research group right now. In recent years, I mean, in the last 2 decades, there's been an increasingly popular discourse about the data deluge. And I think no one has been more vocal on this issue than the rarely quoted scholar or singer, Frank Zappa. Frank Zappa in 1978, in his Packard Goose song, sang, "Information is not knowledge, knowledge is not wisdom, and music is the best." The first part of this quote reflects a well-known conceptualization of knowledge, which goes from data to information to knowledge to wisdom. Today, the focus is obviously on data as the most elemental or the most fundamental unit of knowledge. You can see popular magazines with covers like this one, The Economist, talking about the data deluge and how to handle it, which is at the same time a threat, but also something which can be harvested and can make fertile grounds or make this wonderful flower blossom. You can read in this magazine and others about the data deluge, about data storms, about data tsunamis, data floods, who threaten to drown those who have not learned to swim in a sea of data. This conversation has been most often heard in the context of social media and the amount of data they collect about ourselves and the issues of privacy, or in the context of national security, like here in the United States. But of course, this debate has also been very prominent in the sciences. This cover of Nature talks about science in the petabyte era and this arrival of big data. Popular Science drew some of the consequences of this new age about data, which is power, and a new way in which people who have more power, who have more data, are more powerful. Wired magazine had a particularly interesting take on this story. They argued that this rise of this new data deluge meant nothing else than the end of science. The subtitle here reads, "The quest for knowledge used to begin with grand theories, but now it begins with massive amounts of data." What they were referring to is a new way of doing science, which they called data science or data-driven science. This new way of doing sciences was not completely new, of course. It's been already talked about in the previous decades, for example, in the context of genomics. Here, Pat Brown and David Botstein, molecular biologists at Princeton, were writing in Nature Genetics that this new science of genomics was about collecting and analyzing data to discover things we neither knew nor expected and see relationships and connections among the elements, not testing theories and models. You can see how comparison here, even though it's not explicitly mentioned, plays a crucially important role. The very concept of data-intensive research or data-driven research was theorized to some extent in this book, The Fourth Paradigm, which came out in 2009. It's edited by Tony Hay, which you might not know, but he's the vice director of research for Microsoft. The concept, the word data-intensive scientific discovery and fourth paradigm had been coined by Jim Gray, a computer scientist from the Silicon Valley, who was actually lost at sea shortly before the book came out, and the book is dedicated to him. Jim Gray had coined this term fourth paradigm after the experimental, the theoretical, and the computational paradigm. Then came this fourth paradigm, which is this data-driven science, in reaction to the massive production of data in genomics and metagenomics by another sailor, Craig Venter, of course, who was one of the authors of the Human Genome Project. This view of science as being dominated by data-driven science had not only supporters, it had also critiques, like this cell biologist, John Allen. And please don't ask me why they all put pictures of them either on a ship or underwater on their website. So John Allen here was he is a cell biologist, and he said it makes absolutely no sense to be promoting this kind of data-driven science. It's like asking for the epistemological equivalent of a perpetual motion machine. It simply cannot work. If you don't have a hypothesis, it's just like testing the effect of Italian opera on yeast. It makes absolutely no sense. And Google, who knows so much about how we think, actually also has an answer to this question. If you type data-driven science, the second line which comes there is data-driven science is a failure of the imagination. So you can see that this notion of science as data-driven science is being hotly debated even today. But I think pros and cons might agree on this definition of data-driven science. It's a science that is most explicitly and exclusively devoted to the ordering of complex data. I'll tell you in a few slides who actually coined this definition. But what everybody agrees on this story is that this is new. This is a new way of doing science, a way in which collecting, comparing, and computing are absolutely central. I take inspiration as a historian, of course, from other sources and other scholars who try to question the novelty, this novelty. And Robert Darnton, the book historian, for example, wrote in 2000 that every age, in a way, was an age of information, but each in its own way. And I found this far more inspiring and interesting to question in what ways are we living in an information age, or in what ways is our data deluge unique to the 21st century. I've learned a lot and tremendously from Anne Blair's book, for example, on Renaissance data deluge in her wonderful book, Too Much to Know. In this book, she looks at how scholars at the time were overwhelmed by amounts of written documents and how they literally cut slices of books and paper and sorted them in these wonderful cabinets by themes, a way of comparing and collecting all kinds of information in order to produce new knowledge. I also find extremely inspiring to read about early modern and Renaissance natural history. For example, The Science of Describing of Brian Ogilvy, who writes about this incredible moment when European naturalists discover plants and organisms of the New World, and suddenly the amount of plants and animals that they know of grows by almost an order of magnitude, and how they start to organize and make sense of these organisms and comparing them to others in this engraving from Dürer, which you might know. In the 17th century, Leibniz was also complaining about this amount of data or of information. He was talking here about this horrible mass of books, which is always growing, in which he felt obliged to read all of them. Some of the technologies and techniques to deal with this data deluge of the time were, for example, the encyclopedia. The Diderot d'Alembert encyclopedia is also it's a technology to organize this amount of data, to make it manageable, and to be able to compare one chapter and another from different volumes, for example. And it's not just Diderot d'Alembert, but it's encyclopédistes, Pankouk and the others of that period. Jumping fast forward into the 20th century, the documentalist Paul Otlet from Belgium was writing here about this enormous mass of documents accumulating in the past, which is growing every day, every hour of new units in numbers which are disconcerting and sometimes just outright scary. Of these new documents, like the water falling from the sky, we can say that it can provoke floods and a deluge, or it can fertilize and irrigate. So you can see this ambivalence about this amount of information, which is threatening, but also a promise for producing new knowledge. Paul Otlet, like others, imagined all kinds of machines and devices to deal with this information. Some kind of proto-computer here based on cards and index cards here. So perhaps we can conclude here briefly this talk to say that actually what's happening today is not new. We could critique the novelty of what's happening today. I think a more interesting question that we could ask is rather in what sense or how is what is happening today new? What is actually new from today, knowing that some elements are not new, but some others might be. And this is why I'd like to insert this very broad conversation into a more specialized and specific conversation, which is about the history of the life sciences as one field in which this evolution of epistemologies or epistemological ways of thinking about the world has changed. For those of you who are not historians of biology or historians of the life sciences, I'm going to give you a history of biology over 3 centuries in just about, I think, 3 minutes. I'll try to keep it down to 3 minutes. So I'm going to cut some corners for sure, and I'm probably going to infuriate all historians of biology out there. The history of biology, until very recently, has been often told as a story of 2 traditions, 2 main traditions. The first tradition is, of course, the naturalist tradition, the tradition of naturalists who, from the Renaissance and the early modern period, were mostly concerned with collecting items and bringing them in a central place, a wonder cabinet, like here in Naples in 1599, Ferrante Imperator's cabinet, and comparing these different elements and trying to make sense of them. This tradition is wonderfully described in this very inspiring book by Paula Finlan, Possessing Nature, about museums and collections and the culture of collecting in early modern Italy. It wasn't only Italy, it was also Germany, here in the Wunder Cabinet, also Paris, here in Buffon's cabinet of the king. He was the curator, if you want, of this cabinet. And when you know Buffon and his a towering figure in the history of natural history, he wrote all these volumes of Histoire naturelle. People usually forget the subtitle. The Histoire naturelle is a description of nature, but it's also, and perhaps mostly, a description of the king's cabinet. So this practice of collecting objects and bringing them in a single place where you could compare them, you can see them all within a single, just in one site, was a way to produce new knowledge and organize this knowledge, something that was then became called Histoire naturelle. Buffon was, of course, not the only one to work this way. Cuvier at the museum in Paris was also collecting different skeletons and fossils. And this is, of course, explains in part the rise of museums in the 19th century, which are focused on bringing into a single place all these different items in order to be able to compare them. This was not just the big institutions, the big museums of Berlin, the Museum for Natural History in Paris, but also in New York or in Berlin and elsewhere, but also the private cabinets of Alfred Wallace, for example, co-author of a theory of natural selection here. This was his own cabinet. He had all these drawers, this kind of mini museum in which he had butterflies, of course, but shells also, and all kinds of other organisms. This is also the tradition you find so prominently in botanical gardens. Botanical gardens who collect all kinds of specimens, bring them into a certain place, organize them, and keep them in these loose sheets, which you can reorganize. If you want to compare different plants, you can always bring them together on your own table. And in a single site, you can see plants which never lived together. You can bring together a plant from Madagascar and a plant from Bavaria and have them and look at them at the same time, which is a key practice for comparison. This practice of natural history continues to the present day, obviously, in museums, especially like here in the Smithsonian Museum of Natural History in Washington, DC. I think it would be a mistake to think of this tradition as organismic biology. Because it wasn't only about organisms, although it was often about organisms. It was also about bones, about skins, about fossils, and about different parts of organisms. I think what characterizes best this tradition is that it's a tradition of collecting, of comparing, of computing to produce knowledge. The key epistemic activity here is, of course, comparison or comparing, which is, I think, of interest to your group. If you want to sketch in one slide the historical evolution of this tradition, I'm going to do it right here without no data whatsoever. And it pretty much looks like this. It rises tremendously in the 19th century. It starts to decline around 1900, and it really starts declining quick in the 20th century and becomes less and less important within the life sciences in the mid-20th century. Now, when historians of biology tell the story, they tell this story, or they tell another story, which is the story of this second tradition, which is called often experimentalism or experimentation, which starts about the same time at the Renaissance, here with Harvey making experiments on blood circulation. but it's really a minor tradition, which is at the time, which is really dominated by natural history. And it really rises only in the 19th century with figures like Koch, like Pasteur, like Claude Bernard, all these experimentalists who really make biology and the life sciences or the experimental medicine experimental in a way. And they focus on experimentation. From the 19th century, experimentation becomes scientific modernity in the life sciences, like it did half a century earlier in physics or in chemistry. So in the 20th century, biology or the life sciences and the modernity of the life sciences became equated with the laboratory and experimentation. This is so obvious when you look at movies from the early 20th century. Look at Frankenstein, which takes place in part in a laboratory, which is the place that raises all these great fears about experimentation, the laboratory, but also the great hopes in this other movie, Dr. Arrowsmith, which takes place at the Rockefeller Institute in New York, where Arrowsmith here, Dr. Arrowsmith, is discovering new drugs in the laboratory at a time when you could still smoke in the laboratory. In the 20th century, modern biology became equated with experimental biology and experimentation. This became so obvious with the work of Francis Crick and James Watson, for example, drawing from experimental data produced by X-ray crystallography, not exactly by their group, but by Rosalind Franklin. It is so true also of François Jacob and Jacques Monod in Paris here, doing experiments in bacterial genetics, also getting the Nobel Prize here in 1965. Here in the 1960s, the results of experimental biology became the symbol for modern science. And this becomes nowhere less true than in the early 21st century with the deciphering of the human genome, which is really a triumph of experimental biology. This equation between doing science and experimenting became so strong that it was possible in 1988 for Fred Sanger, who got 2 Nobel Prizes for sequencing techniques, to write this: doing for a scientist implies doing experiments. If you don't do experiments, you're not even a scientist. So this is a story pretty much that historians of biology have told in a very, very sketchy way. There is one specific difference which I need to allude to, which is very different from the history, of course, of physics or chemistry, is the problem of organism and individual organism and model organism. Model organisms played a very important role in the history of biology because that is where experimentalists worked. They worked only on a very small subset of the huge diversity of nature. So in comparison to naturalists who were interested in pretty much every possible living organism, experimental biologists were only interested in a few, like mice here. But they believed strongly, like Jacques Monod and François Jacob formulated it, that what is true of E. coli, the small bacteria, must also be true of elephants, which is kind of nice because you don't need to work on elephants and you can just work on E. coli in your lab. So this belief that you could work just on one organism and then that would be true for all organisms meant that you did not need to compare. There was no need for comparison because it was necessarily true for Jacques Monod and François Jacob that something found in a model organism like E. coli was true for all other living organisms. Comparison is not necessary. It's interesting to see how one of the great historians of biologists and biologist himself, naturalist himself, Ernst Mayr, wrote about this in 1982. He wrote about this comparative method and this experimental method as being entirely distinct in biology. The experimental method of Jacques Monod and François Jacob and Watson and Crick and all the experimentalists, and the comparative method of the naturalist, like himself, an evolutionary biologist. He knew or didn't know, he probably knew because he just knew so much, that Auguste Comte had already made this distinction more than a century earlier. In 1338, Auguste Comte was distinguishing the comparative method of biologists and the experimental method of physicists. What's interesting is that he thought at the time that biologists only did comparison because they were all naturalists at the time. And he was partially right. He was not a great historian, as we all know. But half a century later, biologists also became experimentalists, as I tried to show you earlier. So if you want to put this in a sketch here, so you have this story of natural history, of this naturalist tradition, and you have the rise of experimentalism, which rises from the 19th century and becomes dominant in the 20th century. And today, in the 21st century, big data arrives here. So this is the narrative, if you want, or the background narratives, which were in place when I started writing this book, which became Collecting Experiments. And I thought that this background narrative was very, very unhelpful to understand what was going on in contemporary biology, and especially in this big data biology. And my attempt in this book is to tell a different story. I wanted to tell a different story of the rise of what became called this data-driven science or contemporary life sciences and biomedical sciences. And I wanted to see that in a different light. So I started thinking again about, of course, natural history, present natural history. And I wondered why we thought what was going on in the Smithsonian was so different than what is going on in molecular biology labs at the MIT and Cambridge and elsewhere. Why do, when people collect here sequences of DNA, data about gene expressions here in microarrays, or structures of proteins, when they align these carefully and compare them systematically and try to order them in different categories and build taxonomies from them, isn't this also a practice of collecting, of comparing, of computing to produce knowledge? Isn't this a practice that is centered around collections, like we had wonder cabinets? Just today, we call them databases. Isn't this a new kind of comparative science in other ways? Another way to look at that was to look at where biological biodiversity is. If you ask anyone, where is biodiversity? People would say, of course, in natural history museums. Indeed, the American Museum of Natural History has 500,000 species. But GenBank today, this collection of DNA sequences and RNA sequences, has data about almost as many species today. So the locus of biodiversity here is as much in the Great Natural History Museum as it is in the great data banks of genomics today. So when you think again of this citation, which I used to describe the current era of biomedicine, the science that is most explicitly and exclusively devoted to the ordering of complex data, it's important to realize that this quote is not from the 21st century. It's from 1961, from J.J. Simpson, the great paleontologist who was writing about his practice of collecting, comparing, and computing fossils at the American Museum of Natural History and elsewhere. So the fact that this same definition works so well for these two very different traditions, which we think completely separately, is something that I found inspiring to think of these traditions together rather than separately. And it also helps to explain why molecular biologists or here crystallographers like Jane Richardson in the late 20th century can write about her practice of describing molecular structures in this way, about this vast accumulation of information about protein structures that provides a fresh opportunity to do descriptive natural history, as though we had been preserved with presented with the tropical jungles of a totally new planet. So even she thinks of herself as doing a new kind of descriptive natural history. So the argument of the book is that you have to think together of this comparative and this experimental tradition rather than oppose them, as most historians of biology had been doing up to that time. So in a way, I was thinking of hybridization, another very biological concept, with the corn in the background. That's a private joke for biologists. So in a way, instead of telling this story of these two different traditions, one collapsing and the other one rising and starting to dominate the first one, I thought it would be better to think of course, the rise of experimentalists, which goes without question, but looking that the naturalist tradition has been rising at the same time in the 20th century. And big data is something that happens at the conjunction of this naturalist and experimentalist tradition. So pretty much that's the argument of the book. That's what I tell in the book. And now I'm going to just tell you a few words about the chapters, a brief overview of the chapters, which has many other wonderful stories. Historians love to tell stories. Some of them are boring. I think they're not here. I think there's lots of really exciting stories, which are just fascinating in their own, just for what they are here. So the different past I wrote about in chapter 1 was about live museums, museums of living things. And I start with the American Museum of Natural History, which in the framework of this tuberculosis exhibition in 1908 starts to have collections of live bacteria. And these collections of live bacteria eventually become the stock collections in which on which genetics grows in the 20th century. Here's the collections of seeds of corn, for example, at Cornell. collections of living mice here in Maine with all kinds of mutants. You can see these boxes in the background where you have different types of mice. Collections of flies, Drosophila here at Cold Spring Harbor, at Columbia University and Caltech, which are being organized and collected here, and all the way to the frozen museums of, again, back at the Museum of Natural History, of DNA and other bodily samples. The second chapter and looks at the blood banks. So collections not of live organisms, but of parts of organisms, especially of blood. And I look at how naturalists of the early 20th century, the very early 20th century, like George Nuttall in Cambridge, UK, was collecting blood from all kinds of organisms and trying to use experimental methods of serology and immunology today to classify all these organisms. So doing natural history, but on an experimental basis. He was having all these test tubes and this special rack to put test tubes and compare how the blood of all these different organisms compared and could be compared serologically. In the United States, Alan Boyden was continuing this tradition, and he built a serological museum of blood. And I love this picture because you see these two university officials on the right who are coming and look at the museum, and they're looking at the box, and they look puzzled because there's really nothing to see there. You just see these little plates of blood, and they all look the same. But it was the same tradition of displaying and showing these different items, which were representative of different organisms. Boyden also developed machines, and here he has his assistant who was developing a machine to perform comparisons between these different samples, following this ideal of mechanical objectivity. And he was working on all kinds of organisms. He was fishing lobsters here in Maine and elsewhere, and having this lab put into a bus to travel through different regions, including in Africa, to collect bloods from all kinds of species. The newspapers, local newspapers, were very puzzled by this enterprise, and they call it the strangest blood bank. So at the same time, a blood bank and a blood museum. Charles Sibley, the ornithologist here in Papua New Guinea, this is actually in the 1940s, there's a mistake here during the war. He was also interested in continuing this with birds and collecting albumin from eggs, but also blood from all kinds of birds. He was on a ship, on a laboratory, put on a ship here and going through different islands and trying to collect all kinds of blood from organisms. He would use the latest experimental methods in the 50s and 60s and 70s, like electrophoresis, comparing profiles of proteins, representative, which he thought, of different birds, and then comparing that in huge atlases. The next chapter looks at other atlases, and especially the tradition of sequencing proteins, of looking at the sequence of the proteins of organisms and trying to do natural history from that. It starts in Sweden with Margaret and Birger Blombach, who are doing comparative biochemistry, a tradition which is very important from the 1930s to the 60s, but about which almost nobody has written about. And this is the tradition of comparing biochemistry and biochemical processes in very different organisms. This chapter starts there, continues with Margaret Dayhoff, a fascinating character, a physical chemist in the United States, who for the first person to start collecting systematically all kinds of sequences of protein, sequences and structure, and puts them together in an atlas for the public in 1965. The atlas is tremendously boring. It contains pages and pages of sequences of different proteins and one reference to the person or the group who sequenced it. She invented interesting fold-outs where you can have aligned different sequences of proteins so you can see just within one site these different sequences and compare them visually. She also put all that on punch cards, which was a storage media for early computers, and from which you would at the same time compare sequences, but also produce the atlas. And she would also try to visualize these with all kinds of models, like these little beads on like a necklace here. She was also using the latest computers of the day. Here's a representative on the right from IBM who's bringing a new computer to the president of the lab where she works, Robert Ledley, who is the person who did the most to bring computers in biomedicine in the 20th century. And she also advertised here on German television here in a staged interview about this new method of using experimental results to explore ideas about evolution. Then I look at virtual collections, the kind of collections of proteins that were built in Cambridge by John Kedrew and Max Perutz, for which they got the Nobel Prize. These awfully cumbersome models, 3D models, about which several historians have written wonderful texts, especially Soraya Chadarevian. And I'm looking at here how different crystallographers and computer scientists have tried to make these virtual, and bringing the new technology of computers from the MIT and Brookhaven to make virtual models that look like that you could handle in all kinds of ways. And how these people work with crystallographers like Helen Berman in the United States, trying to collect all this data about all these different structures of protein, which they want to describe, collect, and compare systematically. And how they were infused with the ideals of the counterculture of the late 60s and early 70s, which included ideas about sharing as much as you could, everything you had, including data. This brings to the fifth chapter about public databases and how the first databases of genomic data were made public on a national scale in the United States here at Los Alamos. How a physicist could start to be the emblem of data sharing after working on things like the H-bomb at Los Alamos, something he omitted from his CV, by the way. And how these places, which were some of the places which were connected to the early ARPANET, became the places which became models for diffusing through networks, computer networks, information, in this case, genomic information. And how these people started to conceptualize the link between users and consumers of data, and thought that they were always linked and had to work together, and curators, I'm sorry. And how others like Margot Dayhoff was working with more primitive, if you want, technologies like modems. You see a telephone modem here being hooked onto a computer. So from your home in the late 1970s, you could call the database of Margot Dayhoff and look up her data and compare it. And then look at how this story actually builds the momentum for open science, and how many of the same people who are involved in these new databases are the ones who are going to be pushing open science in the 1980s and 90s. Because they realized that the rise of all these new comparative methods, computerized methods, will require openness on the part of the database curators, openness which was not common at the time. So I look at the career of Richard Roberts and others who have been pushing this vision of open science all the way to the present. And looking on this postcard, how they have been trying to bring together those who produce data and those who consume data. That's why you have this circle around the computer where all the data is being stored. Now, for the last part of my talk, the last 10 minutes, I'd like to say a little bit why I think this is important. And this is the things I addressed in much more detail in the book. And this is about the tensions that I see everywhere in the 20th century when these two traditions come together. And this is particularly obvious today when you read Science and Nature and you read about collections and databases and privacy and the ownership of data. You see these tensions flare everywhere. And I believe that the way in which I tell the story of collections and comparisons and experimentations helps us understand these different tensions and also find some solutions for them. My intellectual resource here was moral economy. So E.P. Thompson's idea here about what is the right exchange, what is the value of things that you exchange, and what counts as a correct moral economy here in exchange. He was thinking of food riots in Europe at a very different time period. But I think it's a very productive notion to think about what was going on today. What I think is going on in the 20th century, and especially in the late 20th century and early 21st century, is a reinvention of the reward structure in the experimental sciences. It just cannot work as it did before because there are just too many tensions coming when it starts to hybridize with this naturalist tradition. These tensions occur in three different fields, and I'm just going to have one or two slides on each one of them. It's about ownership of data. It's about authorship, who gets to put his or her name on something, and about privacy. So first, a few words about ownership. In the natural history tradition, collections were mostly a commons. It was something that belonged to everyone and neither to the collector nor to the individuals who had collected every specific item. So not to the person who had collected that parrot or that bird, nor to the director of the museum who had all these in his or her collection. but they were a commons. Ernst Mayr was writing in his Methods and Principles of Systematic Zoology in '53, a specimen used as a basis for description or illustration becomes common property, belonging to science rather than to an individual. In the experimental sciences, things were very, very different. Data, in this case, or experimental results, were private. They belonged to individuals. You can see this in many places, but I like here J.D. Watson, who's writing, Jim Watson writing his autobiography in 1968, and he tells about how he stole Rosie here, Rosalind Franklin's data. So he says, I was more aware of Rosie's data than she realized. Rosie, of course, did not directly give us her data. And people who reviewed her book went as far as said that Jim Watson and Francis Crick actually stole Rosalind Franklin's data. So you can see that all these people, whether they agree with Jim Watson or not, share the idea that data is something that is owned. And so you can see that there's a completely different idea of ownership here, which is at play in experimentation, in the experimental tradition and the natural tradition. So when both come together, you can expect, of course, clashes. And this is exactly what you see in Nature in 2002 here, a journalist writing about this string of clashes between those who collect and those who interpret data. For example, one story among many, this guy here in the picture, he's at Woods Hole, the United States. He sequenced the genome of a bacteria and then realized that some other group had actually analyzed that data and published an analysis of this data. So he thought he was being scooped with his own data instead of having somebody else replicate his data. And this made no sense and then raised a big debate about who owns a genome once it's sequenced. The second issue is about authorship, which is closely linked to ownership. Ernst Mayr again, he says in the same book, giving proper credit is one of the most important ethical responsibilities of the science. Credit should be given to the collector, the person who collects all kinds of specimens, for example. And this is absolutely clear when you read any book of natural history in the 19th or 18th century. This book, I just took a random book of the Botany of the Antarctic Voyage here by Joseph Hooker. Of course, he did not collect all the specimens which are described there. He collected a few of them, but he is the one, the single author in this book. When another huge work of collection is being published in the 21st century, for example, the human genome, you see something very different. Every single individual who contributed something has his or her own name there. There are 3,000 other names on the internet which follow actually these which are just printed here. So you see completely different ideas about the collective ideas of authorship and individual authors of individual ideas about authorship. And finally, privacy. Privacy is something that we discuss a lot in terms of our own lives and social media, but is also something that is being extremely debated with the privacy of data and the data stored in databases. And I like just to show you this tension here, an editorial of editor of nucleic acids research who writes in 1989. Scientists would like to access everyone else's data, though they do not necessarily wish to reciprocate. So here's just another example of how someone is acknowledging that there is some tensions and something that is not going easily in this new world, this hybrid world, I call it, of experimentation and natural history, if you want. So in conclusion, I'd just like to reiterate the main point. I believe there are strong tensions still going on, which are still unresolved between different moral economies. I believe what's interesting, which is going on today, it's a reimagining of these moral economies in the comparative experimental traditions or sciences and how they become hybridized today. These are very creative ways that involve all kinds of new devices, some of which you're familiar with, I'm sure. Open science is one way to deal with this tension, making all experimental data accessible. Data authorship is something else, being able to claim authorship, not just for analyzing data or performing experiments, but for actually producing data. That's why you have plenty of journals which are being published today, where you can publish just data and have your name attached to it and to your CV. And finally, public participation as another way in which these tensions are being handled by bringing the public in to compare, but also to compare this massive amount of data and analyze this massive amount of data. With that, I'd like to thank you all for your attention. And I'm looking forward to your questions. Thank you.